Dear students, this is Dr. Pooja Thakkar, a biology expert. Here we are in the fourth session of cell biology, that is endomembrane system. Endomembrane system is basically consisting of varieties of cell organelles. The membranous system has been initially formed from the nucleus. The nuclear membrane starts forming more and more components in them, and then that will lead to formation of multiple structures from the same. That will lead to formation of endoplasmic reticulum first, likewise the Golgi body, then the lysosome, as well as the same membrane is also getting occupied for the formation of vacuoles. So the overall membrane is of same kind, which is present at the outermost covering, likewise as the nuclear membrane, and to different components of the cell. So the overall system is called endomembrane system, that is inside membranous compartments which are interlinked to each other, and they are also playing a very significant role for the production as well as transportation of various molecules. Here in this particular diagram you will be able to notice the nuclear membrane is forming the rough as well as smooth endoplasmic reticulum. The tubules from that will play a significant role in the formation of Golgi body. The vesicles which are formed from the Golgi body will be considered as lysosome, and then the same kind of membrane is also present as the name tonoplast, which is the boundary of a vacuole. Now, when we talk about the first component of endomembrane system, that is endoplasmic reticulum, it has been previously studied by Cloud, and the name was given by Potter. This is the tubular structure, as you can see in this particular diagram, also the red and orange color structure. So this is the system of membranes of all the eukaryotic cells except the RBC. It consists of nearly 60% of the total cell membrane, as it is super larger and bigger in size. It is often connected with the outer membrane of the nuclear envelope, as we have discussed. The yellow boundary of nucleus is associated with the red or orange boundary of the endoplasmic reticulum. Now these particular ER has three components or three compartments. The first one is called cisternae, that is the sac-like structure or tube-like structure, which are interconnected and they are parallelly located. They are typically flat in size and shape. The next thing is called tubule, which is the branched network. Okay, so the branched network will be typically forming the tubule-like structure, and at the end vesicles are there which are round and oval structure, which has been pinched off by having the various products like proteins or lipids. Now when we talk about the types of endoplasmic reticulum, the endoplasmic reticulum bears chromosome on its surface. If they are present, if ribosomes are present, then that kind of endoplasmic reticulum is called RER or rough endoplasmic reticulum, which is mainly involved in protein synthesis as well as its secretion. These kind of RER is abundant in the cells which are active in or engaged in active secretion process of the proteins. Fine. But in the opposite case, smooth endoplasmic reticulum does not bear any ribosomes on its surface. So SER is preliminary involved in the lipid synthesis as well as the production of steroidal hormones. Okay, so it is basically present in the cells which are originally engaged in production of vitamins as well as varieties of carbohydrates. So basically SER plays a very significant role in detoxification of the body. SER is having more tubules as well as vesicles. They are not at all having more cisternae in them. In muscle cell, if we talk about, the sarcoplasmic reticulum is present, because the endoplasmic reticulum which is present in these kinds of muscle cell are typically called sarcoplasmic reticulum, in which we are maintaining the proper concentration of calcium for the muscle contraction process. So when the calcium ion releases, that will lead to contraction of the muscles. New endoplasmic reticulum may develop from the existing one, or it can be produced from the nuclear envelope as we have discussed earlier, or from the ordinary membrane of the cell. So anything can be the source of production of endoplasmic reticulum. Now in this particular diagram also you will be able to notice the nucleus, nuclear pore, and the nuclear membrane through which the particular ER is attached. Either it is rough or smooth. Smooth endoplasmic reticulum is very smooth in shape, but they are rough in shape in case of rough endoplasmic reticulum, that is rough boundary bearing individuals. Now the next important component is Golgi body, which has been first observed by Camillo Golgi in 1898 near the nucleus. Nearly, you can say nearly present in between the nucleus and the outermost boundary, near the endoplasmic reticulum, you might say. They are present in all the kinds of eukaryotic cell except the RBC and sieve tube elements, which are typically present in plants. They consist of many flat-like disc-like structure, parallel sac called cisternae, same as in the case of ER, the very big network of tubule as well as the vesicles. The components are basically labeled same in case of ER as well as in Golgi. A unit of Golgi is called Golgi-some. In plants, these kind of isolated Golgi units are called dictyosomes. But in animal, they can form single compact unit or they can form loose complexes depending upon the requirement of that particular cell, or the cell is engaged in which particular kind of process. Now here is the diagram of Golgi apparatus. It is basically having two phases. As we will discuss in the next slide, that more Golgi body are present in that particular cells which are basically engaged in secretory activity, as well as the cell which is involved in cell division process. Now as we have shown, as I have shown in the diagram, that it is having two phases. You can see in the diagram, that is cis phase and trans phase, right? So cis phase is the convex phase, or it can be called forming phase, where the material has been received from the endoplasmic reticulum. And then the trans phase, as you can see, which is on the cytoplasmic side, not at all towards the ER side, which is a concave and maturing phase towards the plasma membrane, that will release the material in the form of vesicle. So Golgi body is mainly serving as the site for the modification as well as packaging of material, and they are storing the material till they are required. Then they are involved in the or engaged in the transportation process. So for example, you can say in the very easy manner that a protein is synthesized typically in the ER, that has been transported through the vesicle using the cis phase into the Golgi body. Then they are modified. Now which kind of modification here in the Golgi body it has took place? The protein has certain kinds of extra amino acids which are not at all required for the functionality of that particular protein. So those extra amino acid has been removed first. Then the protein will become mature, fine, because it is having the particular sequence of amino acid which is required. Then if the particular protein is needed, the glucose moiety or the sugar moiety, that has been introduced there and get attached to the protein. So now that particular protein will be called as glycoprotein. That protein has to be more and more modified, stored, and then they are transported using the vesicle towards the plasma membrane. As we have seen in the content of plasma membrane, those are glycoproteins and glycolipids which are present on the exterior boundary, and they are helping in cell adhesion processes. So these kind of glycoproteins as well as varieties of glycolipids has been constructed in Golgi body in the very mature manner, and they have been transported to the specific location for the target function. Now single membrane bounded structure or vesicular structure present in the cell is basically basically formed from the packaging of Golgi bodies are called lysosomes. They are basically rich in, or they can be having 60 different kinds of acid hydrolysis enzymes, typically nucleases, lipases, proteases, carbohydratases, phosphatases, and other enzymes for the digestion of various materials. And they can also helpful in digestion of all the kinds of organic substances into the animal body except cellulose. So they are most abundant in typically WBC as well as osteoclast. Now what do you mean by osteoclast? Osteoclast are bone deforming cells. they are digesting the bone minerals. And osteoblasts are bone forming cells. They'll work on the backside of this osteoclast to form or to reform the bone. Okay, so WBC and osteoclast are basically degrading kinds of cells, and those kinds of cells will possess more and more amount of lysosome, and these lysosomes will play important role in solubilization of material which is obtained from phagocytosis as well as pinocytosis. So whatever the waste material has been introduced in to the lysosome, that has been introduced into these kinds of cells, WBC and osteoclast, has been digested completely by the lysosome present in them. Okay, now they can show varieties of polymorphism, that is the shape change structure, or morphologically they can look or they can show various structures, different structures. As a primary lysosome, like the newly formed uh, typical vesicle, or they can be served as the storage granule. But when we talk about heterolysosome, then it will be a combination of lysosome as well as phagosome. So it can also be considered as phagolysosome. So whatever material has been phagocytized in the cell, that has been lysed by this lysosome. Or they can be of the third type, that is residual lysosome, that is a lysosome having undigested material which is not at all getting digested in that area. Or they can be of fourth kind, that is autophagic. So they are mainly involved in the own cell destruction. Whatever cell in which they are present, they'll digest the overall cell in which they are present. Fine. So that there can be varieties of lysosomal structures which are having the lipid bilayer at the boundary. In the outermost membrane is there the single unit membrane, into which varieties of hydrolytic enzymes are there, which is basically a mixture. And they are having certain kinds of transport proteins also into the membrane for the exchange of varieties of molecules across the membrane. And the last component of this endomembrane system is vacuole, which is the non-cytoplasmic sac-like structure. From the cytoplasm, it has been separated, and that is covered by using a single membrane called tonoplast. Now, this particular kind of vacuole can be of varieties of type. The sap vacuole, first, that is filled with water dissolved organic as well as inorganic material, which is typically surrounded by tonoplast, as we told. These kinds of plant cells have single central large vacuole, but in the case of animal cell, they are present numerous, but they are very small in size, so they are having small sap vacuoles. The next category is called contractile vacuole, which is typically present in freshwater individuals like amoeba as well as paramecium, and they are mainly performing the task called excretion and osmoregulation. Now, what do you mean by osmoregulation? Regulation is maintenance, and osmosis maintenance or osmosis regulation is called osmoregulation. So they'll maintain the electrolyte as well as water concentration inside the cell as compared to the outermost environment. Now the food vacuole, that is the complex phagosome and the lysosome, that will perform digestion. Fine, as I have discussed earlier, that the phagosome is the vesicle which is having phagocytized material, food material, which has been digested by lysosome, and that has been stored in food vacuole. And the last category is gas vacuole, or it can also be called air vacuole, which is found in some prokaryotic individual to store metabolic gases that will take part typically in buoyancy maintenance as well as several regulation processes. So this is all about endomembrane system. It is serving as the main site for the production of protein, production of lipids, production of varieties of steroid hormone, and certain regulatory activities. Kindly like the video and subscribe the channel. Thank you so much. For the registration in the classes, kindly contact on the phone number 990-980-4647. Thank you.